Tan-ta-dan! Welcome to today's episode of Student Rocking the Floor and Teacher Getting Shocked. Well, you will definitely get to know who is who really soon. But before that, let me tell you the reason why I am starting off the session like this. Because you all go to schools, you all might be going to different coachings as well, and coming back from all these places with different emotion. Some might be happy, some might be sad because nothing good happened in school or whatever is there. So this is your reminder to not be sad anymore, to be happy, because after watching this session you are going to gain some information, and this information is going to make you a step closer to what you're dreaming for. That surety I can give you, and you have to give me that surety that you are going to shock me. Okay, and for that, not a bad shock, okay, good shock. For that, you have to rock, my dear children. How do you rock? By showing your consistency, by studying really hard, okay, by balancing play and also studies everything. And if at all you are in, comment down in the comment section right now that I am going to rock PU2 examination. Now, let me tell the story of these two people right over here. So this handsome person that you see is none other than Thomas Hunt Morgan, my dear children, who went ahead and tried to prove chromosomal theory of inheritance. Now the teacher was working on the experiment and the student made the most utilization of this particular time. His name is Alfred Sturtevant. Okay, Alfred Sturtevant saw that, okay, the teacher is experiment, but I should also try to do something. Okay, and he came up with something which is known as gene mapping, about which you are going to study in today's session. So, you see, it's really great. Do you all wait for the teacher to end their story and then continue with what you were doing? No. If that's the case, you should not be doing that. My dear children, the teacher works hard, okay, but you also have to equally put in your efforts and try to find as many things as possible. For example, I might teach you a topic in the next session also, but I want you guys to be prepared for that topic beforehand. You can probably read the NCERT and come for the lecture. It's really good, right? So this is what I want from you guys. Small things, that's all. Now going ahead, not talking a lot, okay, we will be seeing the whole story in detail in today's session and going ahead and seeing what all are the topics we're going to cover. We're going to cover linkage and recombination and also sex determination in organism, two of the most important topics from where you get so many questions. Now this chapter, as I always say, is not something that you can take lightly. It gives you 13 marks in your PU2 and 4 to 5 questions in your KCET constantly. So do not miss this chapter, and if at all you want to download the notes, please join our WhatsApp channel. And if you have not subscribed to the channel, subscribe to it right away. So today I'm super energetic because of my children who commented, or who gave the answers for all the homework questions I gave. So we have Manasa L, who beautifully, okay, put it down all the homework questions one by one. Kudos to you, all of the answers that you gave is correct. Same goes with Prajwal also. I have put 2 questions, okay, there is one more answer that he gave. Kudos to you, Prajwal. And Gayatri, after so many days, she is back. Welcome back, Gayatri. Thank you for answering to the homework questions. You are doing absolutely great. And Sagar Anmol Chopra, very consistent, okay, consistently answering, and he put down a comment which I was really happy to read, and that is, ma'am, I scored 34 out of 35 in first quarterly. I am really happy and super proud of you, and not just you, all of the children who have been working really hard. Your hard work is going to pay off. Now those of you who are watching this video for the first time, I will be asking homework questions in the mid session so that I know that you're listening to me carefully, and you have to let me know the answers in the comment section below. Okay, so now going ahead with what we have studied in the previous session. So I thought I'll add this up. It's a new thing. It's just a small video, okay, nothing much. So over here, my dear children, starting off with the first thing that we discussed, what dihybrid cross. And because of dihybrid cross, what did we get? Mendel put down the law of independent assortment after doing the dihybrid cross experiment. Okay, and then something went wrong in his story. So what went wrong? We discussed all about it. So after giving the laws, then something went wrong. Everything was sad. But then, my dear children, came what? A twist. okay, after what, 37 years, what was the test? 3 scientists came up to the story, okay, and they rediscovered Mendel's work in the year 1900. Please remember the name of the scientist. And further, since microscope and everything was developed, two other people also came in, whose name was Sutton and Bovary in the year 1902, and they understood that chromosomes, okay, that some chromosomes are present in the nucleus, and Mendel spoke about factors, that factors segregate, they separate. Similarly, the chromosomes are also separating. So probably these factors that Mendel spoke about might be on the chromosome itself, was discovered by these guys up here. And they put up a theory for it, which was known as what? The chromosomal theory of inheritance, my dear children. It was majorly put down by Sutton, but yes. Now, please read the theory. I have explained it properly in the previous session. If at all you need to watch it, you can watch it. And obviously, no one will believe you as such. You have to experimentally prove that, and that's where our guy comes in. And who is our guy, my dear children? Our guy is none other than, where is that guy? Yes, none other than Thomas Hunt Morgan. So Thomas Hunt Morgan was the person who was experimentally going to prove what? The chromosomal theory of inheritance. And for that, he used fruit flies. So if at all you have not seen fruit flies, they look almost like this. So why Thomas Hunt Morgan loved fruit flies? That is the first question. So by this, I try to mean that why did he choose the fruit flies? Why did he choose the fruit flies for his experiment? That's the question. So the reason why he chose fruit fly for the experiment goes on like this. First thing is that they're very easy to grow. Now this is a 3-marker, 2-marker question that you can get. What are the features of fruit fly which made it a perfect experimental material? So first one, they grow on very simple media. Okay, they don't say like, I want fancy stuff. No. And they have a very short life cycle that goes up to 2 weeks, just 2 weeks. So basically, if at all I made some changes or I want to look at the results, within 2 weeks, I could get the results. Okay, that is the use of shorter lifespan. Now, high progeny count per mating. So obviously, while he was doing his experiments, he needed to see, so he needed a lot of specimens out there, a lot of organisms out there. So basically, these, the fruit flies which are there, whenever they mated, they gave rise to so many progenies, babies. Okay, and clear male and female distinction was there. Fun fact, you have to remember this because this is a KCET question as well. By the meaning of male and female distinction, I mean that I can look at the organism and say which is female and which is male. So the one which will be larger, okay, it will be female. Okay, and the one which is smaller, it is actually the male. So this is very contradictory than what we see normally, right? So the larger one is female and the smaller one is male. I'm not going in depth. There are so many other changes as well. But at least this thing is something that you have to remember. Now, visible hereditary variation under low power microscope. So even under low power microscope, he could be examining certain changes. Okay, certain variations which were there, he could clearly see that under the low power microscope, which was actually a good point. Okay, which was also equally a good point. So how many points do you have in total? 1, 2, 3, 4, 5. First one, simple media. It can be grown on simple media, shorter life cycle, that is just 2 weeks, high progeny count, okay, and clear male and female distinction, and then visible hereditary variations under the low power microscopes. And now one very important thing, fruit fly is a name that we gave them, okay, that is a nickname. What is the scientific name? It is known as Drosophila melanogaster. What is it? It is Drosophila melanogaster. Okay, Drosophila melanogaster is none other than the fruit fly itself, and this was the experimental material for proving what? The chromosomal theory of inheritance. Who was doing this experiment? Thomas Hunt Morgan and also his friends, and also he had some students as well. Now going ahead, did he find something else while proving chromosomal theory of inheritance? Well, the answer to this question is yes. He found something that is known as linkage and recombination. That was something very new. So he was trying to prove that chromosomes have the genes, and whenever chromosomes separate, the genes also get separated. That is what he wanted to prove. But then while he was doing the experiment, he got these two things as well, and I am going to tell you what do you mean by linkage and recombination right over here. So this is one of the most confusing thing out there, but trust me, it's very, very easy. Okay, so now we are gonna decode linkage. So what is linkage? Physical association of genes on chromosome is called linkage. Now that would be so much confusing for you, so I'll make it very, very easy. Look, forget this diagram, okay? I've got an easier way to explain you this. So suppose you have A gene over here and small b gene over here, okay? And suppose you have, what do you call, capital A gene over here and capital B gene over here. Fine. Now what happened is that these two chromosomes, when they came together, some swapping happened. Okay, some parts got swapped. This went here, this went here. Okay, now what is the new combination that you will get? You will still have capital A and capital B gene together, and again you will still have small a and small b gene together. It just got swapped. Why like that? Why both of them went together? Because, my dear children, these two genes that you see over here, or these two genes that you see over here, okay, they are what? Tightly linked. They are what? Tightly linked. They're very close to each other. On the other hand, suppose it was something like this. There is small a, there is small b, and there is capital A and there is capital B. Now if I think of swapping one portion of it, the lower portion, okay, what will happen? Here I have small a, here I would be having big B because it got swapped. Similarly, here I would be having capital A, and here I would be having small b because it also got swapped. Now, my dear children, if I see, I get a new combination out there. Why? Because the genes which are there, they are far away from each other, and this is what I call as what? Loosely linked genes. So I have two type of gene. One is tightly linked and the other one is what? Loosely linked genes. And this physical association of genes which you see, my dear children, this is known as linkage. So it can be either tightly linked like this, or it can be loosely linked like this. Now, ma'am, what's the use of them being tightly linked and loosely linked? The answer to this question is that basically over here, as you clearly saw, you can find a new combination, right? Small a was never with capital B, but since the swapping happened, now you see that it is with capital B. I find a new combination. And what do you call this process wherever you get a new combination? This is known as recombination, or new combinations, recombining to form a what? A new combination. And why is it important? Suppose you had some genes which were like this and that, it was very normal, very toxic gene. Okay, it was not a good gene. But now, since the genes are swapped somewhere here and there, you get a new combination and probably new character as well, which could be helping you really, really well. Which is why recombination is good at times. Recombination can be bad, but it can be mostly good at times as well. That is the importance of recombination, and this is what Thomas Hunt Morgan found. That is, Mendel said, according to the independent assortment principle, my dear children, there are two genes, okay, they are on two different chromosomes, both of them independently assort to each what you call gametes. But suppose the genes which are there, they are on the same chromosome. And in that particular case, can they independently assort of each other? Do you think they can? Small a and capital small b gene, can they independently assort of each other? The answer would be no. Why? Because they are tightly linked. They are tightly linked. So while Thomas Hunt Morgan was doing his experiment, he found linkage and recombination, which totally said that law of independent assortment is not always correct. There are cases where the genes can be tightly linked and it might show linkage and they might not assort independently. So if at all a question comes, that is, linkage is against what? Linkage and recombination is against what? So it would be against what? The third law of Mendel. The third law of Mendel. So he was working on what do you call the chromosomal theory of inheritance, talking about the chromosomes and the genes. All of it was absolutely correct. But he found something else, and that was this, my dear children. So over here, I hope now you understand what is linkage. Linkage is nothing but the physical association of genes. It can be the tightly linked or it can be loosely linked. And now we come to recombination. Recombination is what? Generation of non-parental generation of non-parental combination. That means something that you didn't see in the parents. Now this combination that you see over here was not seen in the parents. Parents were small a, small b, capital A, capital P. Over here you're seeing a new combination altogether. So this generation of newer combination or non-parental combination is known as recombination. And when will recombination be high? This is a very important question. Whenever the genes are loosely linked, that's when, whenever the genes are far away from each other, that's when recombination would be high because they can move away. If they are closely linked or tightly linked, the rate of recombination would be what? Less. So I'll write it separately so that you remember it always. Whenever you say tightly linked, that means these genes are very close to each other. Okay, very less distance, write it somewhere. Less distance and also lesser recombination. But on the other hand, whenever I say loosely linked, that means there is small a, small b, capital A, capital B. There is enough distance, large distance. And whenever you have so much distance, what happens? There could be more amount of recombination. I'll tell you an easy method of learning this. You and your best friend. If at all you're very close to each other, you will not allow the entry of a third person in your group itself. Okay, that means no new combination out there. You are the example of tightly linked. Now you and another best friend of yours, okay, who is fake best friend. Who is what? Fake best friend. In that case, my dear children, you guys are not actually close, which is why what will happen? There is a chance that other person can also get into this particular group. This is what? Higher recombination, higher chances of recombination. I hope now you can understand this perfectly. Now, who gave this? It was Thomas Hunt Morgan who gave all this thing. Now, while he was doing his experiment on things like that, okay, so let's decode this. So over here, my dear children, you have this very, very important picture which blows away everyone's mind. But it's easy. It's not very, very difficult. And to make it easy, you have to learn some very important things. So over here, as you can see, there is some small y, small w, y plus, w plus. What does that indicate? You need to know that first. So small y gene indicates yellow body. Okay, what does it indicate? Yellow body. Small w means white-eyed. Okay, white eye condition is known as small w. Now over here, y plus means instead of yellow body, they would be having brown body. And W plus means instead of having what you call white eye, they will be having red eye, my dear children. Well, you need to understand one very important thing, that is the plus sign indicates that they are dominant. Okay, so these characters are the one that are dominant. What is dominant over here? It would be brown body and also red eye. That is something which is dominant, and that is written as wild type. Wild type means they would be dominant over here. So here, as you can see, both of them are X chromosome, and this chromosomes that you see are X and Y. The Y chromosome is not carrying anything, it's just the X chromosome which is carrying something. So over here it is mentioned this is male and this is female. So female has two X chromosome having these two genes, okay, and male has XY chromosome having these two genes. I hope that's clear. Okay, now I'll write it since you might forget it and it might be confusing for you, so I do not want you to get confused. Listen, this is yellow bodied, okay, and the W represents what? That they have white eyes. So yellow body with white eyes, imagine like that. Similarly over here, they have brown body and eyes are what color? Red color. Now my dear children, let's cross them. Okay, we have to cross them. So over here, from the female, one X can come, and from the male, X can come and Y can come. Either of them can come. Suppose this came, okay, and along with that came this particular X from the male. Fine, from here one X came, either of the X's would be same because they have the same genes. So over here one of these came, From here you got one of this. Similarly, my dear children, there is Y chromosome. Y chromosome is here. From here again another one might have come. Whatever you do, what could you see over here? The result would be what? There could be a female being produced, but showing the dominant character. Why? Because you got one chromosome from here which had the dominant alleles, which is why they will show the dominant character, that is wild type character. That means over here it will be what? Brown bodied and red eye color. Now similarly, if we talk about males, since they just have Y chromosome and Y chromosome do not have any alleles, the only alleles which are there you get from the mother itself, okay, that is the female, and female were having the recessive character yellow and white, so which is why over here the progeny will be yellow and white, yellow body with white eye color. Got it? Now you got the progenies, one male, one female. What are you going to do? This is the F1 generation, okay. Now what are you going to do? You are going to cross them. Now when you are going to cross the progenies, you will get some parental type progenies. That means that is just like the parents, either wild type or yellow and white. And you can also see one more very important thing, that is what? Recombinant types. That means new non-parental type progenies as well. Remember one very important thing over here, the genes that you see, they are tightly linked. So do you think they will show high amount of recombination? The answer would be no. Whenever it's tightly linked, it will show lesser recombination, and by lesser recombination the percentage is written 98.7 of them are just like their parents, only 1.3% of them are like what are the new ones which are out there. So now we are going to see what are the parental types. So my dear children, how do you make that combination? I'm just decoding this, okay, so that it is going to become easy for you. So over here, again from this chromosome one guy can come in. Let's suppose the blue one came in, the blue X chromosome came in. So the blue X chromosome is here. From this again one can come in. Let's suppose Y chromosome came in. So over here you have this and this, okay. Which character will it show? As you can see, Y plus is there, W plus, which is why it will be what? Brown bodied and red eye color, the wild type. Now the next combination that can be formed is the upper one, the yellow chromosome can come from here. Again the Y chromosome can come from here. I'm talking about the males first and then I'll talk about the females. So both of them are males, one having this from the mother's side, other having this from the mother's side. Either way over here, since there is no one to dominate, it will be what? Yellow bodied, white eye color. Fine. So these are the two things. Now let's finish this side first and then we'll go to that side. Fine. So over here again from the female side, if I talk about the females, again this chromosome can come and also this chromosome can come. From female side this chromosome can come. From male side that chromosome would be coming in X. That is how you get the females, right? So here X, there X, so you got XX. And now the combination, if you see my dear children, since there is a dominant allele, it will be wild type itself. Now again, since this is gonna come and this is gonna come, both of them are having the recessive allele, which is why the recessive character will be shown. But anyway, they are just like their parents. There is no new combination being seen over here. Now if you see the new combination which is up there, how does the new combination actually happen? So for the new combination to happen, you need to understand that probably these two chromosomes, that is from the mother side, X and X chromosome were resting, their exchange, some parts of them might have got exchanged. So how did I get this new chromosome out here? So you have to use your tiny little brain over here. So suppose my dear children, this blue part that you see, from here some parts got exchanged to this. So it would be having Y plus and now small w. Where is W plus going to go? It is going to go at the upside. Okay, to the upper side it will go. So there is an exchange over here, okay, between the W plus and the W, my dear children. This goes upwards and that comes downwards, which is why you have a new combination altogether that looks like this. Capital Y and small w. And this is the chromosome that you got, okay. So here recombination might have happened, here here crossing over might have happened, which is why you got a new type of X chromosome, and from here you got the Y chromosome, my dear children, which is like this. Now obviously it is going to show a new character. What is it? Y plus shows what? Brown body, but this time brown body is with what? White eye. Okay, white eye. Instead of with red eye, it is now with white eye. Similarly over here also, again probably, okay, so this went down, this went upright. Now we are talking about the other chromosome where the exchange took place. Now it would be small y and W plus, my dear children, up at the top, right? Because they got exchanged. So that is written over here. Again with the male chromosome, that is the Y chromosome right over here. Here also you find a new combination, that is what? Yellow body with what? With red eye color. W plus stands for what? Red eye color. That is a new combination that you got. Similarly, in the case of female also the same thing happens. Suppose here the exchange have taken place between the alleles. Then, my dear children, there will be Y plus and small w, and the lower one will be the same itself. There is no exchange over here. Okay, there is no genes, right, to get exchange. So, it will remain like this. Just a second. Yeah, it remains like this. Okay, and over here again, since there is one to dominate, okay, it will be brown bodied but with white eye color. Similarly, over here, this chromosome remains the same, that is from the mother's end, because there is another Y chromosome. There is no chance for exchanging stuff, right? And from here exchange will be happening, the other exchange material would be coming with this, and you get this combination. So either way, my dear children, you see very less amount of recombination is, very less amount of recombination is present on this side. Why? Because probably the genes might have been closely linked or tightly linked. This is what Thomas Hunt Morgan was trying to prove, and this is what I am also trying to prove you. Why I am teaching you all this stuff? Because in your NCERT one very important line is mentioned, and I want you to understand that, and for understanding that you need to understand this. So I see what percent of recombination, only 1.3% of recombination between these two genes, because they are closely linked. Now if you go over here, the type of recombination, the range of recombination is going to increase. Why? Because here the distance is more. The distance is what? More. So white eye and miniature wings. So it is a structure of wings. It is miniature wings. So over here W plus is there, that means red eye is there, and red-eyed Drosophila with what? M plus. M plus is normal wings. So white with miniature wings and red-eyed ones with what? With normal wings out there. So this is the combination that we are having. Again, my dear children, suppose we are fusing each of them. Whatever you do, the same chromosomes remain the same here. This chromosome can go with this. Okay, whatever the chromosomes are gonna come same from the female. So what small w, small m, and over here the blue one can come once. And the next combination would be what? The same thing plus the Y chromosome which is up there. So you got this. Okay, you got this particular thing. Both are like parents only, right? That is the F1 progeny. Now if I suppose I cross the progenies, I am going to find so much amount of recombination. How much percentage of recombination? 37.2% recombination, my dear children. Why? Because they are far away from each other, which I can find so much different types of recombination right over here. So if I do just like that, okay, for just making you understand things, I take one chromosome from here and the second chromosome from this parent's end. Let's make the males first. And then again one chromosome from here, the upper one, and one chromosome from here, the Y chromosome. I got the males. These are just like the parents. Now similarly, for making the females, I'll take one chromosome from here and the upper chromosome, that is the X chromosome. I'm taking that. Similarly this and that. I will get all the parental time, but the parental type combinations are pretty less over here. Only 62.8% of them are like the parents. More of them are recombinant types, because more amount of recombination has taken place. Now how does recombination take place, my dear children? So over here between the X chromosome, exchange might have happened. Here small w plus and M plus were together, but now since there is an exchange, W plus and normal M will be together, and this M plus which is there, it will be going up. There is a new combination being formed. The reason I am not making it is that when I make it, it will zoom out. Okay, so I won't be able to show you this. So there is an exchange over here that has taken place. You imagine like that. Now I have a new combination of chromosome with W plus and normal M. Okay, W plus and normal M. Now this will combine with this, I get a new combination altogether. W plus was what? Brown eye, right? And it is going with miniature wings this time. It had to go with normal wings, but now it's with miniature wings. Why? Because there's an exchange that has taken place. Similarly over here also, there will be normal W with M plus. Normal W with M plus, because the exchange has taken place, and also the Y chromosome. Similarly, you go ahead, you will find new combinations here also. So the recombinant types are more over here. Why? Because, my dear children, the genes are located very far away from each other. And he did this experiment to actually show that yes, there is linkage and recombination happening, and this is the person, that is the person, and things like that. Now, why I explained you all this thing? The reason is simple. You get questions from it. There's a straight question that comes that the genes are there. They show 37.3% recombination. Are the genes tightly linked or are the genes loosely linked? So now since you know that 37.3 is a big amount of recombination, you will understand that only huge amount of recombination happens when the genes are what? Loosely linked. You get it? And they will be having longer distance between them. By loosely linked, I mean what? Longer distance. To confuse you, they can give you a question that 37.3% recombination are shown by loosely linked genes with shorter distance. Can loosely linked genes have shorter distance? No. That is for the tightly linked genes. So over here, what is written in the NCERT and what is important is that Morgan's findings on linkage. Genes are on the same chromosome tend to get inherited together. Genes which are there on the same chromosome, they usually go together because of what? Linkage. And this is what you call says that Mendel was wrong in some sense, not all sense, in some sense. Now, but strength of linkage varies depending on distance between the genes. Tightly linked genes very close on chromosome, very low recombination. Example, white and yellow genes. So here, white body and yellow genes, they are very close to each other, so they are going to show very less amount of recombination, only 1.3%, whereas loosely linked genes farther apart on the chromosome, higher recombination. Example, white and miniature. They are very far away from each other, which is why they show more amount of recombination. What is the percentage? 37.2% recombination, not 3, 37.2% recombination. So what is the conclusion of this? The closer the genes, the stronger linkage and fewer the recombinants. Now here I give you the first homework question and you have to tell me. Okay, the first homework question, if you understood what I tried to teach you over here, is that if I am saying that is 37.2% recombination between A and B gene or whatever thing is there, my dear children, and you are getting options. Okay, the options goes like this. They are tightly linked. Over here also they are tightly linked. Here they are loosely linked. Don't worry, the options are not over. I have to write more. Loosely linked. Over here they have short distance. Loosely linked and short distance. Over here they have loosely linked and larger distance. Again over here short distance. And this will be large distance. Now these are the options, you have to tell me the answer. For two genes which shows 37.2% recombination, what would be the correct answer? A, B, C, D. Okay, put it down in the comment section right now. Got it? And now, so Thomas Hunt Morgan gave all this thing. And as I said, now it's time for getting shocked. Okay, why? Because Alfred Sturtevant, seeing that all the findings that were done by Thomas Hunt Morgan, he went in and he checked more. What more could be known? Okay, he went ahead and he checked that, and that's when he found out gene mapping. Basically, what do you mean by gene mapping? Please remember, it was Alfred Sturtevant and no one else who put down gene mapping. So what do you mean by gene mapping is that basically with the frequency of recombination, okay, with the percentage of recombination being known, suppose I know that is 37.2% recombination. I know this, since I know this particular thing that is 37.2% recombination is there, I also would be understanding that this much distance probably would also be there between these two genes, which is why these, this big amount of recombination is happening. In simple terms, since I know that these genes are located far away from each other, which is why 37.2% recombination is there, I am actually able to map the genes. I am able to understand that where are the genes on the chromosome. Since they are showing more amount of recombination, I will be understanding they are far away on the chromosome and I will be able to understand the location of these genes on the chromosome, and this is known as gene mapping. what do you call this? Gene mapping. And it was put down by Alfred Sturtevant, and it's very, very important. Why? Because my dear children, in our body, what happens, there are small mutations in certain genes which causes diseases like cancer, thalassemia, a lot of other things. If we know the location of each genes, what will happen? We can change it, even though it's a very time-taking procedure. But if we do it, my dear children, we will be able to cure ourselves. Okay, it's a very advanced thing in what you call in the field of genetics that he did. So that is what you mean by gene mapping. There's a proper definition as well. Alfred Sturtevant used the frequency of recombination between the gene pairs on the same chromosome as a measure of the distance between the genes and mapped their location. Again, reading it out. Use the frequency of recombination, okay, between the gene pairs on the same chromosome as a measure of what? Distance between the genes, and mapped their location right over here. This is known as gene mapping. I hope that is also crystal clear. Now, there's a homework question that I want to give forward, but before that, before I go ahead, put it down in the comment section if at all you have some doubt, because I know this, what you call concept is kind of like this, or you are someone who understood everything really well. In that case also, until this particular point, if you understood things, put it down in the comment section that ma'am, linkage and recombination pakka clear. So if at all you get a question on tightly linked, loosely linked genes, I should be sure that you could be able to answer it. If at all you get a question on who discovered gene mapping, I should be sure you would be able to answer it. It is Alfred Sturtevant. Okay, if at all you get a question who put down linkage and recombination, you should answer it. It is what? Thomas Hunt Morgan. And you should also answer why he chose what? Drosophila melanogaster for his experiments. If you know the answer for all these questions that I have asked, put it down in the comment section that I understood. What do you mean by linkage and recombination? Now, a homework question for you guys. This is homework number 2. Since I already gave one homework, recognize the figure and find out the correct matching. So there is 2 Drosophila being mentioned. So what is it? Is it male Drosophila, female, it's male butterfly, female butterfly, whatever thing is there, you have to put it down in the comment section. This came in KCET. Okay, please put it down. And going ahead with the next thing out there, the next major topic, sex determination in organisms. Okay, I was just looking at time. Okay, so sex determination in organisms is another very interesting topic. And to start with, I have to talk about one guy out there whose picture I could not find on the internet. So it's the story of what? Hen King. So it was the name of a person. His name was Hen King, my dear children. In the year 1891, he said something that made a lot of sense. So he was working on the sperms and things like that of the insects, and he found that when spermatogenesis occurs, when the sperms are being formed, obviously 4 sperms are being formed at the end, right? He found that 50% of the sperms has some body, X body, okay, X body, and 50% of the sperm did not have this X body. Now people were confused that what is this X body, what is this X body? Later we came to know that this X body is nothing but what? The X chromosome. Do you know how the term X came in? We put no, the factor X, we have to find X. So X is always unknown. This is how it came in. Okay, the story of Hen King. Okay, why there is X chromosome over here? Because he didn't know what is it. So he put it as X body, and later it was renamed to what? The X chromosome out there. So when he did this, he looked down the spermatogenesis procedure in insects. I'm not talking about human beings. In insects such as my dearest grasshopper right over here, my dear children, he noticed that 50% of the sperms carried this X body and 50% of them didn't carry this X body. Now, ma'am, how can that happen? We have studied in males, no? In males, X chromosome will be there in some, and Y chromosome is there in some, in the case of human beings. So over here, that is something very new. Over here, you see another type of sex determination, not XY type of sex determination, but very different type of sex determination, which is known as XO type of sex determination. And I'm going to explain you that right now. So here the females will be having 2 X chromosomes, just like how we have in the case of human beings also. But the males will have just 1 X chromosome. There is no other Y chromosome over here. That space is empty. There is nothing, which is why it is represented by O. Okay, it is represented by O. There is nothing out there. So males will be having just 1 X chromosome, nothing else. So over here, this X chromosome came in, or probably females can only contribute the X chromosomes. So over here in the gametes, in all the gametes that the female produce, it will be XX chromosome itself. In the case of males, some sperms will be produced which will be having X chromosome, some of them will be produced which will be having no chromosome at all. Now what happens further? So suppose this X chromosome and this X chromosome came together, you get XX. This X chromosome and this X chromosome, you still get what? XX. But suppose over here, this X chromosome from the female and over here, this gamete which didn't carry any chromosome came together, what do you get? You get XO condition. You get what? XO condition. And this will turn out to be what? A male. It will turn out to be male. So I'll properly write it so that you understand. In the case of female, they will have XX chromosome. In the case of male, they have XO condition. That means there is no other chromosome out there. So what will be the gametes? The gametes can be X. Over here also one more X. Okay, since the chromosomes segregate, half of them will be having X chromosome, another half will also be having X chromosome since they do not have anything else to contribute. Similarly over here it will be X chromosome and it can be nothing as well. Suppose this and this came together, you get XX, that means it's a female. Suppose this and this came together, you got XO, that is, it's a male. Suppose this and this came together, you get XX, again that is what? A female. And suppose this and this came together, you get XO condition, which means that is a male. 50% chance, 2 of them are female, 2 of them are male, right? So 50% chance is there that a male and a female can be there. This is the case of insects and they show XO type of sex determination. And what is our type of sex determination? We show XY type of sex determination, my dear children. So over here I have written down everything. Listen to me carefully. So the one that we have studied right now is an example of male heterogamety. What do you mean by that? So when the males produce 2 different type of gametes, such as over here, you saw that the females are producing the same type of gametes. Whatever things that they do, it is always X. But in the case of males, it can be either X or O, my dear children. It can be either X or nothing at all. So they are producing 2 different type of gametes out there, which is why this is an example of what? Male heterogamety. Now ma'am, in the case of human beings, okay, in human beings what is happening? There is XX chromosome from the female and XY in the male. When they separate, again the females are producing same type of gametes only, that carries XX, but the male are the ones that produces 2 type of gametes, one is X and the other one is Y. So are we also an example of male heterogamety? The answer is yes. And along with us you have Drosophila as well. Remember Drosophila melanogaster, ma'am, is an insect, but it has what? XY type of sex determination. Please remember, do not club it with what do you call grasshopper. Drosophila melanogaster and human beings, both of us has XY type of sex determination. And here, since males are the one producing 2 different type of gametes, I call this as male heterogamety. Remember this example, XY for Drosophila and human beings, XO type for grasshopper, both of them come under male heterogamety. Now we are going to talk about female heterogamety, where females are going to produce what? 2 different type of gametes, something that is very rare. Where do you see it? In the case of birds. So what happens in the case of birds? Let's see it. So in the case of birds, the male birds are ZZ. Male birds are ZZ, just like how in females we have XX. Similarly, males are ZZ and the females that you see are ZW. Okay, now whatever gametes they produce, they will be having only Z to contribute. But over here in the case of female, it can be either Z or it can be W. Now suppose this and this come together, you get ZZ, again that's a male. Suppose this and this comes together, you get ZW, that's a female. This and this comes together, ZZ, again that's a male. This and this comes together, ZW, and that's a female. 50% chance to be female, 50% chance to be male. But here, females are the one producing 2 different type of gametes, which is why they are example of what? Female heterogamety. So birds are the example of female heterogamety. Please remember Z and W. Okay, Z, W, Z, W. Fine. I hope this is clear. please make sure you're taking a screenshot of this particular thing. If at all you're not planning to download what do you call the PDF, because this is where you get so many questions from in your competitive examination as well. Cool. Now we'll go ahead. We saw this type of sex determination, XO, XY, ZW and everything. Now we have a whole different type of sex determination. Okay, why did I write this? Okay, I was planning to show you everything over here, but I have already showed you. But I have already written down and shown you things, right? So I don't think it's needed anymore. So now, as I was talking about something different, and that is seen in honeybees, my dear children. So they have haplo-diploid sex determination. What is it? Haplo-diploid sex determination, my dear children, and it's very, very interesting. So listen to me carefully, otherwise you might lose the track of it. Okay, so it's seen in honeybees. What happens over here? The female, okay, the normal cell which is there in a female will be diploid, 2N, right? It will be diploid. You have studied that only during gamete formation, meiosis happens, and now you have haploid gametes. So over here, the diploid mother which is there, the cell which is there, the germ cell, it undergoes meiosis. You have haploid gametes. So diploid has 32 number of chromosomes. So haploid will be having what? 16 number of chromosome. I hope that's clear. So you have got the gametes right over here after meiosis. Now my dear children, in the case of males, the thing is that they are not diploid. They are actually haploid in nature. They do not have diploid body, they would be haploid in nature. In that condition, they are already just 16. Can you have it again? No, that won't be making any sense. So basically, rather than undergoing meiosis, they will be undergoing mitosis. So instead of undergoing reductional division, getting reduced more, they think let's have equational division, which is known as mitosis, and they keep the number same itself. That is, they will be producing haploid gametes. So it started haploid, it is undergoing mitosis, not meiosis, and they will still produce what? Haploid gametes. Now my dear children, two things can happen. From the female side, the haploid one can come. From the male side, the haploid one can come. Together when they come, 16 plus 16, what do you get? 32. That's a diploid female. Otherwise, one very important thing can happen, that is, the haploid gamete which was there, it can, what do you call, free itself from fertilization. It won't undergo fertilization. Rather than that, it will just convert directly into what? Male, without fertilization. So this is a unique process that happens, and it is known as parthenogenesis. Remember, it's different from parthenocarpy that you see in plants. This is parthenogenesis, where without fertilization, both of them are happening without fertilization, but there you get fruits, and over here you get male honeybees. So over here, without fertilization, it is transforming into a male, which is why, since fertilization is not happening, the 16 remains to be 16 itself. It does not fuse with anything else. So it is just haploid male. So this is known as haplo-diploid sex determination, where the haploid ones will be always males and the diploid ones will be always females. I hope you got it, and this is what you see in honeybees. Do not get confused between Drosophila and honeybee. Drosophila are just like human beings, showing XY type of sex determination. Over here they have haplo-diploid sex determination being seen. Remember it, okay? Females undergo meiosis, they produce haploid gametes. These haploid gametes, and males also produce what? Gametes, but without meiosis, they undergo mitosis. Okay, once they come together, you get diploid female. It's all fine. Okay, and suppose fertilization has not happened, then it will turn to a male, and this is known as parthenogenesis. Parthenogenesis. Now there is an important statement again given in the NCERT, that is, suppose this is the kid, that is a male kid. Male kid, does it have a father? Do you think that it has a father, my dear children? No, because it developed from the female gamete itself. There is no father. So they do not have father does not have son. But this child would be having grandfather. Where do you got the grandfather from? So this was the female, right? And female was formed of one male out there. Female was formed when one male was there. So basically, my dear children, my dear children, this child which is there will be having grandfather because they have arisen from the female gamete itself, right? The female have got a parent out there, which is why they have a grandfather, and there is grandson also, that is this particular thing, but there is no father-son relation in here. Okay, there is no father-son relation, there is only grandfather-grandson relation. I hope it's clear now. If you didn't get it, I'll read it out for you. This is given in the NCERT. They do not have a father and thus cannot have sons, but have a grandfather and can have grandsons. The reason is simple. Over here, without father, you get the male, without fertilization, male is being formed. But this male, the gamete that you get, okay, so the gamete which turned into a male, my dear children, was produced from a female, and this female had a parent. This female had a parent. So here the female gamete and the male gamete came together, right? So the one that where you get the male gamete from is what? The grandfather. Is what? The grandfather. And the grandfather will have the grandson, which is this. I hope it's clear. Now, okay, and it's also a question that comes. So that is about the sex determination. So these are the different types of sex determination which you need to remember. Please make sure you're remembering it. If at all you have doubts, you can ask down in the comment section. But at least for now, you need to know this. Okay? So I hope everything which I mentioned is all clear to you. So there is XOXY type of sex determination which is coming under male heterogamety, ZW type of sex determination which comes under female heterogamety, that is shown by birds, and then there is haplo-diploid sex determination which is shown by honeybees. And now time for a question. The frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes was explained by. So measure of distance between genes, frequency of recombination, where did we study all this thing? When we studied gene mapping. This is a definition of gene mapping, and who put it down? It was Alfred Sturtevant who put it down. It was not Gregor J. Mendel, it is not Sutton Boveri, it is not T.H. Morgan, it is Alfred Sturtevant. So your answer is B. Now going ahead, match the columns 1 and 2 and choose the correct combination from the options given below. XO type, XY type and ZW. So you have to choose 1 and 2 from here. So XO type is an example of male heterogamety or female. XO is usually male. So male produce 2 type of gametes, right? So A goes with 1, A goes with 1. I have 2 options like that. Now XY, again it's a male that has XY condition. So it is again male heterogamety. So again B goes with 1. So B goes with 1. I just have this option. With that I got my answer. But let's see. ZW. ZW is shown in the case of birds and they show female heterogamety. So your answer is this. This type of questions could be coming up, which is why I thought I should make you solve this. Okay. Now, time for, I think, homework question is going to pop up. Yes. So the homework question that is there, which of the following conditions correctly describes the manner of determining the sex in the given example? This is question number 3. Homework question number 3. Which of the following condition correctly describes the manner of determining the sex in the given example? Homozygous sex chromosome X and X produces male in Drosophila. Does XX produce male in Drosophila? Think of that. XO type of sex determination, male sex in grasshopper. Does XO type gives male sex in grasshopper? Homozygous sex chromosome ZZ determines female sex in birds. Does it? XO condition in humans as found in Klinefelter syndrome determines the female sex. But Klinefelter syndrome is not yet discussed, so you can exclude this option. Still you can find your answer. Okay, so please tell me the answer in the comment section. And the 4th homework question, the last homework question I think, in male heterogametic type of sex determination, males do not produce gametes, male parent produce similar gametes, female parent produce dissimilar gametes, male parent produce dissimilar gametes. Put it down in the comment section. It's all easy and it's all related. You can see 2023 question it is. Just 2 years. Yeah, that's all. So that is how we complete what? Sex determination as well as linkage and recombination. My dear children, please put down your doubts and I will be starting, I will be ending this chapter with one very important topic, and that is diseases or disorders. Make sure you are there in the class. It's a very important class. If you have friends who are confused in this topic, make sure you ask them to also watch the video which is going to come day after tomorrow. Okay, so please watch it. It is going to help you. And if you think this video was helpful, do not forget to hit the like button and also subscribe to the channel and share it with your friends. Wishing you all the best. Please take care of yourselves. Bye-bye.